Bismillah Rahman Rahim, and we start another paper 6, which is a 5090 November 21 paper 6-2, which will help you revise for your paper 6, and I hope this is very helpful to you and helps you in your exams. As we all know, the paper 6 timing is for 1 hour, and everybody must remember that when they're starting this exam. Now, as you look at the first question, as it says, all questions have to be answered, and you must know that there's no choice in this paper. You have to do the entire paper. Question 1, the cells have membranes which can allow molecules to enter and leave the cell. Remember, revise, cells have partially permeable membrane. A student wanted to investigate the movement of glucose and protein molecules through a membrane. He decided to use whisking tubing that acts in a similar way to an actual cell membrane. So that means it's partially permeable as well. He has been given two solutions A and B. So we have two solutions A and another solution B. So in a beaker, he gets two solutions A and B. There's something in it. There's a liquid in it. As it says in the question, it's a solution, so it's not something solid. State which reagents you would use to test for glucose and protein. So for test for glucose, of course, nothing else but Benedict solution. But if you knew that, only then you could be really writing it. So Benedict solution, Benedict's reagents or Benedict. So the only thing was Benedict's, which was important. And for protein, you used biuret reagent. So this is all what you had to write, and you got your 2 marks for that. He labeled 4 glass test tubes to do these tests. State what would you use to label the glass test tubes. Okay, so we used 4 glass test tubes. And what would you use? Well, what would you use? Would a water resistant marker or a pen? So you would use a water resistant pen, or you could have said a wax pencil or a water resistant marker. Basically a water resistant, because solution water shouldn't be washed off with the water. Now, part 2 of the question says, suggest what you would write on each test tube and record this in the table on page 3. Well, because you see, what you have to understand is that you were going to do a glucose test on A and then glucose test B, and then you're going to do a protein test on A, and so you basically had to have 4 test tubes, 1, 2, and this had A and B, and then there would be another A and B, and you would do the glucose test on this, and then the glucose test on this. And then you would do the protein test on this, and then the protein test on this. So, I mean, it said suggest what you would write on each test tube and record it in the table on page 3. Now, this is page 3. So, you give the label GA, glucose A, protein A, glucose B, GB, protein B, PB. So, basically, because on 2 solutions, you were only given 2 solutions A and B. So you had to do the glucose test on A and the protein test on A, and then the glucose test on B. You were only given A and B. So this is how you would label it. Then he says the student completed the test and recorded the resulting colors of the solution in his notebook. Glucose test A, pale blue, orange, pale blue. So glucose test A gave you a pale blue color, right? And the glucose test B gave you an orange color. Sorry. Glucose test B gave you an orange color, right? And the protein test A gave you a pale blue color. And the protein test B gave you a purple color. Now think, when glucose is not present, there's no reducing sugar. It's a blue color. When protein is not present, it's a blue color. If it's orange, that means glucose is present. If you do a glucose test, if it's purple, P for purple, P for protein, P for present. So purple, protein, present. Now in part 3, it says complete the table using observations and your conclusions. Conclusions means, what did you conclude? Was glucose present? Was it not present? Did something happen? Did something not happen? Conclusion. Observation means what you see. I see I see an orange color when I do a Benedict's test. What does that mean? It means reducing sugar is present. Then I do Benedict's test, it gives me a brick red color. So conclusion is that there is more reducing sugar than the orange one. So basically the answer would have been observations: pale blue, no glucose; protein A, pale blue, no protein; glucose in B, orange, so glucose present and purple; protein present. So you had to just fill in the table, but only if you knew these colors, you would only be able to do this question. You couldn't have just done it like that. Then it says the whisking tubing that he used was a hollow flexible tube. He tied a knot in one end of the tube and make it in a test tube shape. He carefully added 15 solution of solution B. So the solution B is inside. This is the solution B. At the inside of the tube through the open end, after checking that no solution was leaking from the knotting end of the tube, he rinsed the outside with distilled water, placed the tube containing solution B into a large glass tube that contained solution A. So outside we have solution A. Though this is outside is solution A. And inside the tubing is the solution B, which is this part. This is the B is inside. So B is inside and A is the outside. He placed this tube containing solution B in a large glass test tube that contained solution A as shown in the diagram. So you've got the A on the outside. This is the A on the outside. Please try to understand this question just like I'm doing this for you in the diagram and doing it with the colors so you can just imagine this, right? Then, as I told you, that you're going to ask you practical details. So, C1 state what piece of apparatus you would use to add 15 centimeter cube of solution B to the whisking. So, it would be a syringe. You could have said a syringe. You could have said a measuring cylinder or a graduated pipette or a burette. So, you must know these names of these, and I'm just going to show you a diagram of all of them in a while. Suggest why it was important to rinse the outside of the whisking tube before placing it in the large test tube containing solution A to ensure that there's no solution B on the outside. When you wipe something, you ensure that nothing on the outside. Like for instance, if I poured some ruabza into a glass and I spilled it on the outside of the glass, then I would clean it with a tissue paper so that there was no ruabza on the outside of the glass. So why did you sort of clean it? Why did you do it? You to ensure that there's no solution B on the outside of the test tube. So solution B is underlined. So in order to, you have to say that you would have sort of said that there's no solution on the outside. If it's underlined solution B, then the solution B word should be there, and only then you're going to get that 1 mark. After 30 minutes, the student removed the whisking tubing from the large test tube. He then tested the solution remaining in the large test tube for glucose and protein. State two variables you should have controlled so that these repeat tests or solution A produce results that were comparable with those in the table. Naturally, same temperature for the Benedicts, um, same time in the hot water, same volume of Benedicts, and the same concentration of Benedicts. When you test for Benedict's test, you realize you have to boil it in the Benedicts, and the temperature should be between 80 and 100. So the same temperature for the Benedicts and then same time in the hot water means that you boiled it for 2 minutes or you boiled it for 3 minutes, then it should have been the same in both the cases. Point is that whenever you are doing these tests, you must use the same volume of the Benedict solution. Of course, I've not used the whole word. Same volume of Benedict solution or the same concentration. Benedict solution is made in a certain way. So if that is not made in the right way, and in the previous one you made the Benedicts and somebody else makes the Benedicts for the next, so same concentration of Benedict solution and the same volume of Benedict solution. From these tests, he concluded that the solution in the glass tube now contained glucose but not protein. Describe and explain what this conclusion and the information in the table show about the permeability of the whisking tubing. Number one, it shows you describe means it is permeable to glucose. Why? Because the glucose was not outside. Why has it? So that means it could move. The glucose was allowed to diffuse out. So permeable to glucose, but not permeable to protein. Not permeable to protein, but did not move out. So not permeable to protein got you another mark. Glucose got you 1 mark. Protein got you another mark. And the fact that this is partially permeable. So very narrow mark scheme point. Permeable to glucose, not permeable to protein. And the fact that it is partially permeable got you your 3 marks. Next part, before the student removed the whisking tubing from the large test tube, he observed that the levels of the solution A and B has changed in the shown in the diagram. Start of investigation after 30 minutes. Describe the changes to the levels of solution A and B. Now, naturally, what has happened? Level of volume in whisking tubing B went up. This has gone up. You can see here, this has gone up. So level of level or the volume in whisking tubing B went up. So B has gone up, and level of volume in large test tube A went down. So you can see the A went down here. This has gone down in A. Because it's a describe, remember describe is no biology, so you've got to just tell me a describe. And then that is the most important thing, is that you keep on coming with a lot of things which you don't understand the question, and then you come up and then you say, oh, I didn't get a good grade. The reason you didn't get a good grade, you're not listening to me. You're not understanding. Describe. Describe is no biology. You're not going to give me explanations like, oh, osmosis is happening and this is happening and diffusion is happening and active transport is happening. That's all biology. If you said the level of volume in B went up, got your mark, or if you said level of volume in A went down, that gets you your 2 marks. Nothing else can get you those 2 marks. No other explanation is going to get you the 2 marks. Suggest which process. Now, here comes biology, and the only answer was osmosis. That's it. You couldn't have written anything else. There was no other answer which was valid. So if you wrote osmosis, you just wrote osmosis, you got a mark. Suggest which process. You don't have to say the process is osmosis. You just said osmosis and you got a mark. Now remember, this is 18 marks. And if you do it very carefully, you should only be losing 2 to 3 marks in wrongly wording the answers. Otherwise, everybody should be getting basically a 15 out of 18 in this. That is your goal. Please remember. Now coming to question number 2, transpiration results in water moving through plant stems. This can be measured using the apparatus shown in the diagram. Of course, the name of this apparatus is a potometer, but just look at it carefully. There's a leaf. There is a leafy plant shoot. This part is inside. This part is inside. This is the leaf. This is the photosynthesizing part. There's a water reservoir. There's a valve here. There's an air bubble here, and there is water inside it. So all this is filled up with water. This is all water here. It's all airtight. There should be no air bubbles in it. And this is where we have only one bubble, and then it dips into this water, and then there's water in this beaker as well. So look at it carefully. The cut end of a leafy plant stem is inserted in the apparatus. As transpiration occurs, water moves up the stem and is absorbed. This causes the air bubble in the apparatus to move along the tube towards the stem. So this air bubble, let me just color that. This air bubble is going to move this way towards the stem. So the air bubble in the apparatus moves along the tube towards the stem. The distance moved by this bubble in a known time is a measure of the rate of transpiration. So what are we assuming? We are assuming that this water is going in here, up here, into the leaf and out of the leaf. So that is why the bubble is moving. This is what your assumption is, that all this water is going into the plant and out of the plant. Then, of course, the a student wanted to investigate the effect of air movement on the rate of transpiration. So, of course, they're studying the rate of transpiration, investigate the effect of air movement. So, they're going to have a fan or something to set up the air movement. So, she set up the apparatus in a room where there was no movement of air. Okay, so there's no movement of air, and left it for 5 minutes before recording any movement of the bubble. Explain why the student left the plant for 5 minutes before starting recording. Well, what is this called? It says allow the plant to equilibrate. Allow the plant to equilibrate. Now, where does this word equilibrate come from? It comes from the word equilibrium. And I always give you an example, like you're doing a 100-meter sprint, and the instructor will ask you to first warm up for that. It's sort of you get accustomed, or you become in equilibrium with the environment. So to come into equilibrium, like you come into the class and I say, okay, everybody settle down, 5 minutes, take out your books, take out your pencil cases. So I'm letting you adjust to the environment. I'm letting you adjust to the new subject that we're going to study in biology. Probably you had a physics class before me, so I will let you get settled down. So that's allowed to come to equilibrium. Now it's a little bit of a tough English. Some of you are good at English, some of you are pathetic, sadly. So allow the plant to equilibrate, or you can say equilibrate, or you can say get used to the new condition. You can even use some other words because alternative words are allowed in this. So allow the plant to equilibrate or get used to new condition. Get used to new conditions, or get used to the new environment, or get used to the new um, environment I could think of. She used the valve to allow water from the reservoir to move the air bubble back to the start of the graduated scale. Now that's called reset the apparatus. She then recorded the position of the bubble every minute for 5 minutes. She then set up an electric fan to blow air gently over the plant leaves. After 5 minutes, she adjusted the air bubbles as it was at the start of the graduation tube. Now, please understand why we're doing this. You see, when we open this tap, this is the tap. When we open this tap, what is going to happen? The water is going to go in and the water is going to push this bubble back to the 0 point. So I'm sure you're all understanding this. If somebody hasn't understood it, then I'm explaining it to them. So after 5 minutes, she adjusted the air bubble so that it was at the start of the graduated scale and recorded the position every minute for 5 minutes. Now this is with the fan and that was without the fan. Most of the results are shown in the table. Now here is the table. So time, 0 no, 0 air. Then 1 minute, then so remember it's after every minute. So 1 minute it has moved 1 centimeter. Then in the air, it has moved 1.3 centimeter. In after another minute, at the second minute, I said it has moved 2.1. So remember, it has moved one, then it has moved another, it's come to 2.1. So basically, you've got to understand what we're talking about. So 1, 2.1, 3.1, 4, and 5. Here it has moved 1.3, 4.4, 5.9, and 7.6. The diagram shows the position of the air bubble at 1 and 2 minutes when the plant was in moving air. 1 and 2 minutes when the plant was in moving air. Right? They've shown you a diagram of it. Here's the diagram. Observe the position of the bubble at 2 minutes and record it in the table. Now, this is the experimental thing. You've got to understand it. They're checking how well do you know your experimental procedures. So you just need to be a little more having a lot of common sense. So if it is saying 1.3, so how was this 1.3? This was 1.3. So here, what is it going to be? It's going to be here. So it's going to be what? 2.8. So this was very easy to understand. 1.3 and here you would be 2.8. So this is what you had to do is that you had to look at this. So this would get you overall the position. This would you write 2.8 and you would get that 1 mark for that. Now at part 3, construct a line graph of the data in the table on the grid below. Plot the data as two lines on the same axis. Join your points with ruled straight lines. Now of course here comes the labeling part. Time should be on the x-axis. Why? Because what have I told you in previous this thing is that it's always in the table. This is always the first column is always the independent variable, and this is always plotted on the x-axis. It is the independent variable. So time slash minutes, that is what you write. You must write the units. If you don't write the units, you don't get a mark. So time on x-axis, and of course labeled with units. Now what was really on the y-axis is the position of air bubble and centimeter. So position of air bubble, and you have to write the whole thing. You have to write the units as well, centimeter. All points you have to plot correctly, and then you had to, so please remember there's 5 marks, so there's something more about it. Usually it's a 4-mark question. So x-axis, I've labeled it 0, 1, 2, 3, 4, and 5. Because if you look at this table, it is 0 to 5. So I've put them all, and the 5 should come right at the end of the grid so that we have used most of the grid. So the scale that I've chosen on the y-axis is 0 to 468 because the maximum reading is 7.6. So I'm sort of being a little careful and I have get it down to 8. And So you have to label the every 2 centimeter, you have to label it, you have to choose the scale, you should choose as much of the scale as possible. In A levels, of course, the scale is given by Cambridge, and you have to be very careful about that. Here, of course, no scale has been given. So 02468. So I've done the first one and I've plotted the points. They might not be exactly correct because I can't get it very right. And then you just connect them point to point with a ruler and you go through all the points. And of course, you get this first line, which is no air movement. So you have to label that no air movement. So you would make a key of it that this would be the no air movement. Of course, I've used different colors, but you can use something else and you can say, okay, so this is line A, and then you can give me a key and you can say A is no air movement. And then, of course, you can say B and you can use another line for that and you can label that B and give me the one for with air movement. So I have tried to plot these. I know they're not very exact, but yours must be very exact. You must use a very sharp pencil. And then, of course, you connect these and you get your B with air movement. With air movement. So you understand why there are 5 marks for this graph? You see, there's 1 mark was for time on the x-axis and both axes fully labeled with units. Then continuous scale and over half of the grid used for the plot. So I would have got that mark. All points plotted correctly. If I had done that, I'd probably get a mark for that. Straight lines drawn between points plotted, and there is no extrapolation. So you don't extend them. And lines labeled and a key used. So this is what you had to do, is you had to remember that you had to do this in this manner. Then coming to part 4 of the question, it says use the use the data to describe and compare the effect of still air and moving air on the rate of transpiration. And that's 3 marks. And of course, it's simple. Transpiration rate was faster in moving air, but then you have to give me a little bit of use the data to describe. So transpiration was faster in moving air, then bubble moved 7.6 centimeter in the 5 movements, and bubble moved 5 centimeter in the 5 minutes in still air. So when it says use the data, you have to give me and compare, you have to give me reference to the data, you have to refer to the data. So transpiration rate faster in moving air, a bubble moved 7.6 in 5 minutes, or you could have calculated the rate, which would be 1.5, or in bubble moved 5 centimeter in 5 minutes, so that would be 1 centimeter in 1 minute in still air. So you gave me these figures of a moving air and still air, and you got your 3 marks. Then it says calculate how far the bubble moved between 4 and 5 minutes when the plant was in moving air. So that was very easy. 7.6 minus 5.9 is equal to 1.7, and that got you your 1 mark. The area of the cross section of the hollow glass tube containing the air bubble was 0.785 millimeters square. Use the data to calculate the volume of water that was transpired by the plant between 4 and 5 minutes in moving air. Give your answer to 2 decimal places, space for working. So well, basically, what was it? 17. It was 1.17 into 0.785. But why did you do 17? Because it's 1.7 centimeter. So 1.7 centimeter is equal to 17 millimeter. Because here they're giving it to you in millimeter square. So 17 into 0.785, and whatever the answer to that is 13.25 till 13.35 millimeters cube. 13.35 millimeter cube. Then coming to the last part of the question, another student repeated this investigation a week later, but her results were different. Suggest 2 reasons why her results were different. Naturally, maybe if the temperature was not the same, maybe the humidity was not the same, maybe it was a more humid or a less humid day, then maybe she didn't have all the lights on which the other student had. Then the speed of the air movement, maybe the fan was slower or faster. Speed of the air movement was different because the student does it on another day, or the size of the plant or the size of the leaves was different. Size of the plant or size of the leaves could be anything which was different because maybe it wasn't going to be the same shoot. So this was all the 5 points, and you could have given me any two of these. So temperature, humidity, light, speed of the air movement, or the size of the plant, or the species of the plant, even if that could have been different, we don't know if the same species was chosen. The last question, the photograph shows the surface view of a leaf epidermis. Make a large drawing of the two guard cells between the points C and D and the epidermal cells touching the guard cells. So you had to do that. You had to draw a diagram of that. Let's have a look at that. Measure the length of the guard cell that the photomicrograph between points C and D, and you have to write that down. And that was anything between 13 millimeter or 15 millimeter. The actual length of the guard cell is 0.07. Calculate the magnification of the photograph to the nearest whole number. So I'm sure everybody is very clear on the magnification. If you're not, well, you need to look that up on how to calculate the magnification. Now, the answer to that was 18.57 on the, you figured that out. On the calculator it's 18.57, which comes to 18.6, and most of you would have written 18.6. I'm 100%. But it says to the nearest whole number, and that of course is 19. But you didn't read that, I'm sure. If I had, I do check papers many times, and I see there's so many mistakes. 50% or nearly, I would say 70% of my class gets this wrong. So 19 would have been the answer because it said calculate the magnification. But if it didn't say that, then you could have said 18.6 and that would have been fine. Then coming to the drawing, I'm just going to do a drawing to make you all realize the 4 marks that you're going to get for the drawing. Now, please remember, many of you say that you're not very good in fine arts or anything, but it's not in bio, there's no fine arts. It's just something which you just have to see and draw. Now it says guard cells at least 30 millimeter long. So the diagram should be pretty big. It shouldn't be small. You shouldn't make a tiny drawing. You must use the space. The entire space should be used, which is given to you in the exam. Then sharp pencil and continuous lines drawn for guard cells and stoma, and no shading anywhere. Now you see I have not drawn any shading anywhere. Even this slight bit of it here is not correct, but I couldn't really help it. You shouldn't be doing any shading anywhere. Then stoma is the opening which I've drawn here, the stoma, and which is this part, which I'll just trace it again for you. This is the stoma part. So that is drawn, and 6 cells drawn. One, two, three, 4, and 5, and 6. So 6 cells drawn, nuclei shown in all the cells. I've shown the nucleus with my red pencil. And you can see the red is, and I haven't shaded it. And some of you will, of course, do this, but oh my God, that will be killing. That will be a sad day if you do that. So no shading, no shading at all. Please remember, no shading. Even if you're drawing the cells, the nucleuses, you don't shade it, and you draw it to the size which you're seeing. You see, I mean, if you look at the diagram, these are the nucleuses. This is the nucleus here. I'm just going to shade it with the red. This is the nucleus here, this is the nucleus here, this is the nucleus here, this is the nucleus here. So the nucleus is wherever this is visible here, there's one here as well. So you can see that there are 1, 2, 3, 4, 5, 6 nucleuses because there's 6 cells. So be careful when you're doing this, no shading at all, please, and nuclei shown and in the correct position. So thank you once again, and thank you for watching. And I hope this is going to be helpful to you. And I will just show you a little diagram after this, and you can see just 2 slides. Just some names of the laboratory operators. Please pause the video here and have a look at it and remember these names. Some more names. Please look at these and have a look and just remember them. And thank you once again, and thank you for watching. Please do subscribe, and thank you very much.